Today, we're diving into a really strange environmental puzzle. It's all about a chemical ladder, where you'd think climbing is the goal, but it turns out sometimes the real danger is in falling off.
Think about all the stuff in your life. Your couch, your computer, even your clothes. To keep us safe from fire, a lot of it is treated with special chemicals. But when those things get old, when they break down, those chemicals don't just vanish, they wash into our rivers and eventually our oceans. So it really makes you wonder, what happens to them out there?
All right, so let's get specific. The chemicals we're talking about are called brominated flame retardants, or BFRs for short. For decades, they've been like these invisible guardians in our homes, stopping fires before they can start. But what happens when those guardians escape into the wild?
This is really a tale of two different kinds of chemicals. First, you had the legacy BFRs. They worked great, but we found out they were pretty toxic and stuck around in the environment forever. So they were banned. And we got smart, right? We created novel BFRs, specifically designed to be safer replacements. But did we really fix the problem?
To get to the bottom of this, a team of scientists set up shop in the Bohai Sea in China. And this was a huge investigation. I mean, they were tracking 30 different kinds of these flame retardants, both the old guys and the new ones, across 18 different species, from the tiniest plankton all the way up to big predator fish. Okay, before we get to what they found, there's one key idea you need to get: trophic magnification. Just think of it like a chemical ladder. So a little fish eats some plankton, right? It absorbs the chemicals from that plankton. Then a bigger fish comes along and eats a bunch of those little fish. With every step up that food web, that chemical just gets more and more concentrated.
So really, the whole investigation came down to this one simple question for every single chemical they looked at. Does it magnify? Does it climb that food ladder? Or does it dilute? Does it effectively fall off as it moves up?
All right, so after all that work, hundreds of samples later, what did they actually find? Well, this is where things start to get interesting. This is where the real mystery begins.
Now, for most of these chemicals, the answer was, well, pretty much what you'd expect for these kinds of persistent pollutants. No surprises here. They climb that ladder loud and clear. Small traces in the little guys became much bigger concentrations in the top predators.
So the way scientists measure this is with something called a trophic magnification factor, or TMF. Super simple. If the number is bigger than 1, it's building up. And look at these numbers. They're not just bigger than 1. They're way bigger. We're seeing both new and old chemicals becoming nearly 4 times more concentrated at each step. But here's the twist. This is where the whole thing gets weird. Because for some of the most widely used replacement chemicals, the ones that were supposed to be better, the exact opposite was happening. I mean, just look at these TMF values. They're all less than 1. That means their concentration was actually going down as they went up the food web. They were literally falling off the ladder. And we're not just talking about some old, weird chemical. This included 2 of the major new replacements. So this was the big puzzle, right? How is this possible? These chemicals are all pretty similar. They're all made to do the same job. So why on earth would some of them climb the ladder while others just fall right off? Well, the answer, it turns out, has to do with a property we can just call stickiness. The fancy scientific term is lipophilicity. Basically, it's a measure of whether a chemical prefers to hang out in fat or in water. If it loves fat, it's going to stick to an animal's body tissue. And this is where we get this amazing paradox. So check this out. If a chemical is just a little sticky, it gets absorbed no problem and passed right up the food chain. It magnifies. But, and this is the craziest part, if a chemical is super sticky, its molecules are so big and fat-loving that an animal's gut just can't grab onto them. It's too bulky, so it just gets excreted instead of being stored in fat. And that's what leads to dilution. So it all comes down to this Goldilocks zone for stickiness. It's amazing. To really build up in the food web, a chemical can't be too sticky, can't be not sticky enough, it has to be just right. The ones with medium stickiness climb the ladder, but the really sticky ones are just too big to be absorbed, so they fall off.
Okay, so mystery solved. It's all about the Goldilocks level of stickiness. Case closed, right? Well, not so fast, because this raises a much, much bigger question. If these new chemicals are falling off the ladder, does that mean they're actually safe? And the answer to that is kind of terrifying.
The whole thing really comes down to this one single number from the study, 63,000 picograms per gram. Now, what on earth does that mean?
That number was the average amount of all the new flame retardants they found in the marine life. And here is the absolute bombshell. The total concentration of these newer chemicals was 10 times higher than the old banned stuff they were supposed to replace.
So yeah, maybe some of these new chemicals don't climb the ladder, but we are just flooding the ocean with them in absolutely massive amounts. And this uncertainty is something the scientists who did the study are very open about. They even say that the research in this area often gives mixed and even contradictory results.
And that brings us to the real bottom line here. Just because a chemical doesn't biomagnify doesn't mean it's harmless, especially when you dump tons of it into the environment. What this study really proves is that newer is not the same thing as safer. Figuring out chemical risk is just incredibly complicated.
And so that leaves us with this really tough question, doesn't it? This whole investigation solved one puzzle only to reveal a much, much bigger one. If even the chemicals we designed to be better, to be safer, have these completely unpredictable effects, how can we ever really know what's safe for our environment? It makes you wonder, how do we get off this treadmill of just replacing one problem with another?